Assalamualaikum warahmatullahi wabarakatuh. We are from KK4. We will presenting the production of somatotropin from recombinant of E. coli. Somatotropin, also known as human growth hormone HGH, which stimulates growth and cell production in human and animals. Somatotropin also a secondary metabolic product which produced by E. coli using recombinant DNA technology. The process involved cloning the cDNA and the clone gene for expression in E. coli. The usage of somatotropin also commonly used for children with growth hormone deficiency or insufficiency, and it is now currently approved and marketed for enhancing milk production in dairy cattle. E. coli is a gram-negative bacteria with rod-shaped cylinder as 1 to 2 micrometer long and 0.5 micrometer radius. After homogenizing, the median debris size approximately approached to 0.3 to 0.5 micrometer. E. coli also classified as facultative anaerobes. The table here shows the chemical and physical properties of the reactant and product involved in this production.
So now we move on to the next part, economic issues, supply and demand of somatotropin. The global somatotropin market demand in 2020 was around Rm 19.12 billion and expected to reach approximately Rm 31.24 billion by 2026 at a CAGR 8.61% during 2021 to 2026. However, due to the COVID-19 pandemic in year 2020, the global supply market reduced to 4% from previous year. The global market size is expected to reach Rm 1.34 billion by 2026 from Rm 1.05 billion in 2020, which registered 4.2% CAGR during the forecast period. As you can see at the right side, this is the graph of somatotropin supply and demand, the value of demand increasing steadily in each year. In this slide, the table shows the list of main companies that produce somatotropin around the world. Plant capacity for production of somatotropin. Based on the global demand and supply, by the year 2026, the production rate of plant is estimated as below. The value for demand is 4,600 tons per year and for supply is 200 tons per year. Now we need to find the value for the deficient by minus the value of demand with supply. So we got 4,400,000 kg per year. The operation hour is 24 hours and for the maintenance, we estimated around 576 hours per year. Then we can find the operation day, which is 8,184 hours per year. By using that value, our plant capacity is 289 tons per year.
For the production of somatotropin, the recombinant DNA of E. coli is done by the extraction of HGH from pituitary gland. E. coli is cultivated first in seed fermenter before transferred to batch fermenter. The LB medium with glucose and amino nitrate is supplied to batch fermenter and temperature maintained at 37 degrees Celsius at pH 6.75 to obtain optimal growth of E. coli in fermenter. The oxygen is supplied continuously. The wastewater from the batch fermenter is separated by a centrifugal filtration with biomass and cell. After that, the cell wall of E. coli is disrupted by lysis buffer using high pressure homogenizer at 1000 bar to obtain inclusion bodies. Next, cross-flow microfiltration is used to remove the biomass from the inclusion bodies before the purification process. Another centrifugal has been used, which is ultracentrifuge, to purify the inclusion bodies or reduce the contamination during refolding buffer. Refolding buffer, which contains HCl at pH 7, is used to refold the inclusion bodies to recover functional active protein in missing time. An ion exchange chromatography used to further purify the somatotropin by DEAE Cepros with 95% purity. then 0.5 amp now at pH 8.3 used to wash our product from this chromatography. Finally, the last step of for purification with 99% purity is the somatotropin enter the gel filtration chromatography to separate protein based on the molecule size desired. Higher form of protein is removed through the gel filtration.
Environmental and safety issues. On the left side, you can see the figure that shows stream contain waste generated for the whole plant. And on the right side, you can see the table shows the quantity of waste for each component. So the type of waste are exit gas, wastewater, and biomass. For exit gas, the waste treatment is the pollutant to be isolated is first dissolved and microbiologically degraded. For wastewater, the waste treatment is biological methods such as pond technology and anaerobic method that can remove over 95% biological oxygen demand from the wastewater. And biomass, the waste treatment is conventional combustion and cutting-edge thermal depolymerization.
For the material balance, we are looking into the mass balance in the main fermenter and overall plant. For the fermenter, the equation here shows the general mass balance for unsteady state reaction. And since the fermenter is operating in batch mode, hence the flow rate of going in and out of the reactor is not considered. And the fermentation is occurring at 37 degrees Celsius and 1.5 bar with a growth rate of 0.05 per hour, and the results in this table shows that the material balance in the fermenter is balanced. Same goes for the overall mass balance where we consider all the flows in and out of the whole plant and the calculation shown is balanced. The energy balance in the fermenter is calculated by considering the reactant that goes into the fermenter at 25 degrees Celsius and goes out at 37 degrees Celsius. The energy balance is calculated by assuming that the kinetic energy, potential energy, and work done on the system is negligible, and the results for the energy balance shows that it is an exothermic reaction where 105 exponent 3 kilojoule of heat is released per batch.
So the enzyme we used is adenylyl cyclase, and it is under class of lyases which a group elimination to form double bond. And the mechanism for the enzyme reaction occurs for adenylyl cyclase is induced feed mechanism. As you can see on the slide, the adenylyl cyclase characteristic. Metabolic pathway that used to produce somatotropin is intracellular signal transduction pathways. So the first is the activation of G protein coupled receptors. And then the receptor associates with stimulatory G, which composed by alpha, beta, and gamma subunits. And then the binding activates the phosphorylation of GDP to GTP at the G alpha subunit. The G-alpha subunit associates with GTP stimulates the activity of adenylyl cyclase, and the adenylyl cyclase used as catalyst for the conversion of ATP to CAMP. The reaction increases CAMP levels, which activates the protein pKa. It is allowing free catalytic subunit to translocate into the nucleus. The free catalytic phosphorylates and activate the transcription factor CRAB, and allowing it to bind to the CAMP response element CRE in HGH promoter to complete the DNA sequences of somatotropin. And then the CRA modified the expression of the GH. Recombinant DNA technology was used to manufacture mature HGH in E. coli bacterium. For obtaining a plasmid capable of controlling HGH synthesis in E. coli, we use recombinant DNA of E. coli method. The first is the DNA of HGH is extracted from pituitary gland and the mRNA is isolated from the cells. And then the mRNA undergoes the process of reverse transcription with reverse transcriptase. The DNA sequences are cut by KK4 to remove the signal peptide and the first 24 gene. Then the first 24 amino acid nucleotide sequence was chemically generated and derived derived from the known amino acid sequences of HTAs. Then the synthetic gene of the first 24 amino acid and cDNA is ligated with T4 DNA ligase and it will produce recombinant cDNA of HTH. The cDNA is inserted into plasmid PB322 and transfers into a bacterial host shell E. coli K12 strain W3110. And then this recombinant E. coli is then prepared to subculture in LB medium which contain 200 mg1 of ampicillin for a treatment.
The separation part, cross-flow microfiltration S2 is chosen to separate the cell debris biomass due to the low solid content which is 29% in the stream after cell disruption process in high pressure homogenizer. As we know, bioproducts is very sensitive to heat and loss of product to denaturation or degradation. To control the cross flow and control the flux using the pressure difference and the transmembrane pressure, it's calculated. Based on the figure, the feed permeate retented is in the stream 13, 14 and 15 respectively. And also the table here showing the temperature and pressure involved in the cross flow microfiltration S2. This is the table of flow rates and composition and also the table of filter parameter. This is the heat duty involved in S2. Since there is no reaction occur in this process, hence the enthalpy of rate of reaction is not calculated and the energy balance is negative 9 to 9.04 kilojoule per hour.
For the process control, the first unit operation is the batch fermenter, and in the fermenter we want to control the liquid in the batch fermenter to remain at 37 degrees Celsius throughout the reaction by manipulating the inlet flow rate of the cooling water. And if the temperature is higher than 37 degrees Celsius, then the valve will increase its opening, and if the temperature is lower than 37 degrees Celsius, the valve will decrease its opening. Next is the centrifugal pump, where we wanted the pressure in stream 9 to be 5 bar by manipulating the outlet stream of the control valve. And if the if the pressure is higher than 5 bar, the valve opening will be decreased, and if the pressure is lower than 5 bar, the valve opening will be increased. The cooler E2 is an important to cool down the stream from 20 degrees Celsius to 8 degrees Celsius after the cell disruption process is conducted to reduce any denaturation of the somatotropin product. So the temperature of the stream is controlled by increasing or decreasing the inlet flow rate of the cooling water, which is similar to the temperature controller in the batch fermenter. And last but not least, the control variable in the ultracentrifuge is the flow rate in stream 15 to maintain at 88 kg per hour. And we manipulate the inlet stream of the control valve if the flow rate in stream 15 is higher than the set point 88 kg per hour, and then the valve opening will decrease, and vice versa if the flow rate is lower. And this is the overall PNID for the whole plant, where all the control configuration that we taught earlier just now is considered in this PNID.
As conclusion, somatotropin, also known as human growth hormone, is a polypeptide with 22 kDa in molecular weight with 191 amino acid. The method we use for our production is recombinant DNA of E. coli. The plant capacity is 33 kg per hour. The waste of our production is wastewater, acid gas, and biomass. The heat released by the fermenter is negative 111.09 out of 3 kJ per hour. The enzyme used to produce the somatotropin is adenylyl cyclase. For the separation, we use CF microfiltration to separate low biomass in solution. For the controller part, we mainly focus in the batch fermenter, centrifugal pump, cooler, and ultracentrifuge. Lastly, the pressure vessel use batch fermenter and anion exchange chromatography to calculate.
That's all from us. Thank you.